thank you so much, and thank you to CTNNB1, Connect and Cure, as well as the Med13L Foundation for inviting me to be here today. And thank you for so many families who have already visited me, so I have some ongoing speech projects upstairs, and hopefully I'll see a few more of you today and tomorrow. So today I'm going to talk to you about childhood apraxia of speech and dysarthria in rare monogenic disorders. And so what are rare monogenic disorders? CTNNB1 and Med13L, as well as, of course, many others. So I'm going to highlight some of my more kind of larger research in genetic disorders, but I will pull out some of those findings that are particularly relevant for both CTNNB1 as well as Med13L. So an overview, what are motor speech disorders? I suspect that many of you are familiar with them already, but for those who aren't, I will provide a brief overview of those. Then I'm going to talk about a study that came out earlier this year looking at genetic etiologies of motor speech disorders through a large clinical exome sequencing project. And then I'll talk about motor speech disorders specifically in Med13L. And so this is some research that some of you might be familiar with in the Med13L community. I'll talk about a publication and some additional data that I've collected since then. So first, what are motor speech disorders? How does speech happen? For most of us, it's automatic. You know, we don't think about it. We have a thought in our head and it comes out. But there's a lot of steps that happen in between that. So first we have the cognitive linguistic stage. So this is where we decide, number one, that we want to communicate, and then we begin to formulate that message. The second stage is transcoding. So once we know what we want to say, we have to figure out those motor plans that are involved in actually producing the message. After that is execution. So once we have those motor plans, we actually have to execute them. To execute them, we need to coordinate a lot of movements. These are called the articulators, things like the tongue, the lips, the soft palate, the larynx. All of these things have to work together in concert to produce fluent speech. And then lastly, we have sensory feedback, and this is an important part too. So sensory feedback is, there's an auditory component, so how did that message sound? There's also a proprioceptive component, so how did it feel? And when we get this feedback, this helps us kind of tune the message so that the next time we produce it, it's produced more accurately. So for most of this happens in concert. This happens almost instantaneously. We don't have to think about it. But for some kids, this process is really hard. And you can probably imagine then when something breaks down at any single step, it leads to some problems. Those problems are kind of under this umbrella term called children's speech sound disorders. And speech sound disorders basically just mean problems with speech clarity. So these are kids who are really hard to understand. But they fall on a continuum. So on the one hand, we have these higher order cognitive linguistic problems, oftentimes called a phonological disorder. And then on the other end of the spectrum, we have anatomic problems. So these are problems with the physical mechanism that's used to produce speech. So kids who are hearing impaired or have a cleft palate. So problems can occur kind of anywhere in that continuum. So when we're talking about motor speech disorders, we're talking about dysarthria and apraxia. So these are problems that occur when there's a breakdown in motor execution or motor planning specifically. Speech sound disorders in general are really common. It's actually the most common problem of early childhood. It affects up to 5% of all kids between the ages of 3 and 17. But when we look at motor speech disorders specifically, these are actually quite rare. So we can kind of define then motor speech disorders as disorders or problems with articulation that are caused by deficits in either motor planning and programming, leading to apraxia, or motor execution, which leads to dysarthria. And these things actually are comorbid a lot of the time, much more often than we would expect by chance. There's also this third category called speech motor delay or motor speech disorders not otherwise specified. And this is kind of a catch-all term for those kids who really don't fit cleanly into one category or another. So they might have some motor planning problems, some motor execution problems, but don't fit nicely into a category. As I kind of alluded to before, even though speech sound disorders in general are common, motor speech disorders are actually quite rare. So they occur, we don't have really good figures about this, but the study that we usually like to quote says that they occur in about 1 to 2 per 1,000 children. So it's not common at all. We know, however, that motor speech disorders are more common in certain genetic populations, as well as more complex neurodevelopmental disorders. So it's actually really rare. Clinically, I very rarely see a child come in who just has apraxia. There's almost always something going on. Oftentimes that's language problems, it could be autism, it could be global delays, motor impairment, but it's actually quite unusual to get a child with an isolated motor speech disorder. And then there's some good literature showing that certain genetic disorders are associated with a really high penetrance of these motor speech phenotypes. We don't have as much information about MED13L and CTNNB1, but that's one of the questions that I hope to answer by being here. So the first study that I'm going to talk about came out a little earlier this year, and we were looking at a large clinical exome sequencing population of children who had previously been diagnosed with either apraxia or dysarthria, and we wanted to try to figure out the genetic etiologies for these children. So the objectives of that study was to leverage the clinical exome sequencing data for the largest cohort of children with motor speech disorders to date. We wanted to identify new genes associated with MSDs. We wanted to determine whether certain genes are statistically overrepresented among children who have this type of a phenotype compared to just the general neurodevelopmental risk that a lot of these genes do confer when you get a variant in them. We also wanted to evaluate the molecular diagnostic yield of exome sequencing as both a function of sex as well as neurodevelopmental comorbidity. So these were 2,000 children who either had these previous diagnoses of apraxia or dysarthria. They were ages 2 to 21, so included young kids, but also all the way through adolescence. And then they underwent clinical exome sequencing and CNV, or copy number variant detection. So this was actually a sample from GeneDx, which is a large clinical genetic testing company, and we were able to share data with them. We did exclude participants who either had a hearing impairment or were described as having absent speech, as this would kind of preclude a solid diagnosis of an MSD. And then we were further able to kind of pull out some broad phenotypic information to say whether or not they fit into one of these neurodevelopmental categories, so either global delays or intellectual disability, autism, motor impairment, or seizures. So these were kind of broad categories, but we wanted to get a better idea if there was a higher yield of exome sequencing in these particular populations. So we were able to look at the variants, and we evaluated them using standard guidelines to determine whether or not they were pathogenic or considered causative of the phenotype. A second thing that we did was this multivariable logistic regression model to estimate the association between these comorbid neurodevelopmental diagnoses and the likelihood of having a positive genetic finding. And then the other thing we did was something called a gene-based burden analysis. Essentially what this is we wanted to look at the motor speech disorder kids and the genes that kind of shook out in that analysis, and then compare that frequency to the really larger, clinical exome sequencing population of just kids who went and got exome sequencing for any indication. So not specifically motor speech disorders, but just neurodevelopmental disabilities in general. And the reason for this is we really wanted to identify those genes that had the highest likelihood of a specific association with the motor speech phenotype above and beyond just the risk for neurodevelopmental disorders more generally. So the main results, we had a positive diagnostic result in 26.3% of children, so pretty high yield. It was significantly higher for females than for males. And this isn't unique to motor speech disorders. We kind of find this across a lot of neurodevelopmental difficulties, so we do find a higher yield in females more generally. There wasn't a significant difference in the diagnostic yield between apraxia and dysarthria, and that's kind of interesting. So the studies in the past, the genetic studies in the past have looked exclusively at apraxia. To my knowledge, no one's looked at dysarthria, but I think we definitely should, because there's a really high yield there too. And then individuals who had things like autism or global delays were more likely to have a positive genetic finding. So out of the 527 individuals who had a positive finding, about 95% had sequence variants. So these are variants in specific genes, and then the remainder had what's called copy number variants, which are larger deletions or duplications. 262 genes were implicated, so it was a lot. So it wasn't just, shaking out a handful of genes that have an association. It seems like there are a lot of genes that might be implicated in apraxia and dysarthria. Although we will say that 102 genes had 2 or more cases, and there were actually 24 genes that had 5 or more cases. So there really were a handful of genes that had kind of a bigger signal than the others. And then we found 29 unique copy number variants, including quite a few of 16p11.2 deletion, which previously in research we found has been associated pretty significantly with motor speech disorders, so no surprise. So these are just the top 13 genes where we found variants in 6 or more cases, and you'll see that both CTNNB1 and MED13L are represented up here. Another reason why I'm super interested to be at this conference and happy to be working with you guys. One of the things that I did want to highlight is with the majority of genes, we found cases who had both Apraxia and Dysarthria. So there was a lot of overlap, and I think that just kind of speaks to the fact that I think there is some shared pathophysiology there in the two. Even though they're separate disorders, there's clearly something going on where certain genetic variants confer risk for both. However, I do want to say that there was a big difference. CTNNB1 and MED13L were unique in that they both kind of had a signal for either Dysarthria or Apraxia. So MED13L was related a lot more strongly with Apraxia. CTNNB1 was related a lot more with Dysarthria, which I thought was pretty interesting. So we did this gene-based burden analysis. Again, it's a little kind of confusing to describe, but the purpose of that study was to figure out, did we find variants in these genes at a rate higher than chance? So if we just looked at all of the exome sequencing population, agnostic of whether or not they had a motor speech phenotype, would we have found the same genes? And the answer is no, not necessarily. So there were about 30 genes that were overrepresented in our particular cohort, the motor speech cohort, with an odds ratio of between 2 and 2.5 for both MED13L and CTNNB1. Of course, when we corrected for multiple comparisons, which you have to do, that was no longer significant. So there was a signal there, but potentially we didn't have quite a large enough sample size to be able to say for sure. And this graph just kind of shows you the ones that were significant. So there were actually only 3 genes that survived study-wide correction and 2 genes that survived actual exome-wide significance, meaning we compared all of the genes in the genome and used that, and there were still 2 that were significant there. So the conclusions from this study, there was a high genetic diagnostic rate overall, particularly for girls and for those who had comorbid neurodevelopmental disabilities. This was the first study that examined the genetic underpinnings of dysarthria, so we know a little bit more than we did before. We found a similar diagnostic yield between Apraxia and dysarthria, so I think it makes sense to start looking at dysarthria as well. There were a lot of new genes that were implicated in motor speech disorders. And then the biggest take home for this presentation is that both MED13L and CTNNB1 were prevalent findings in the cohort, and they might be overrepresented among individuals with motor speech disorders compared to other children going through exome sequencing. Okay, so now I'm going to switch gears a little bit and talk more about the motor speech phenotype that we know about in MED13L. So some of this work was based on the data that we collected back in the Baltimore mega meeting a couple of years ago, so thank you to all the families here who participated in that study. And then I will also talk a little bit about the paper that we currently have in revision, where we looked at some data from Simon Searchlight as well to kind of combine. So the aim of this study was to characterize the speech, language, motor, cognitive, and adaptive phenotype of individuals with MED13L related disorder. And I kind of alluded before that we had 2 different cohorts. So we had an in-person cohort of 17 individuals, 16 of whom we recruited at that meeting, and another one who I followed clinically, and then the virtual cohort. And the virtual one was the larger Simon Searchlight cohort that I'm sure a lot of you have participated in. So I'm going to, in the interest of time, skip ahead a little bit to the take-home messages. So the in-person, one of the most interesting things we found was there was a really significant kind of speech impairment component. So about 6 individuals used single words, 2 had some phrases, 3 used simple sentences, and then we had 2 children who I tested who were completely conversational. 3 children we found were minimally verbal, and for the purposes of this study, we defined that as fewer than 10 consistent verbal words. 41% used some form of AAC, mostly speech-generating devices, and then 9 reported some type of oral motor dysfunction. So a lot of them, it was difficulty feeding in infancy, drooling into later childhood, open mouth posture, some oral hypotonia. And then the speech phenotype, I think this was the most interesting finding. So everyone, the 14 individuals we were able to test and make a differential diagnosis, met criteria for at least one motor speech disorder. So 7 had apraxia, 6 had both apraxia and dysarthria, and 1 child had just dysarthria. So that's a very high rate of this rare motor speech problem. Looking at the language, cognitive, and motor testing, I won't talk much about this, but basically when we talk about standard scores, 100 we usually say is the average, and then average range goes down to 1 standard deviation. That's that top line there is the bottom half of the average range. And so we found that kids had both receptive and expressive language delays, particularly in vocabulary, which is what we looked at. Verbal IQ was lower than nonverbal IQ, so nonverbal IQ was a relative strength. And then kids also had a pretty significant problem with visual motor integration, so fine motor skills. Looking at the Simons cohort, no big surprise, so most kids, most caregivers reported some degree of cognitive impairment and language impairment, lower rates of other things like autism and ADHD. So language impairment was by far, along with cognitive impairment, the most common feature that was reported by parents. And then with results of the Vineland, you can kind of see that there were delays across the board, but in the orange there, that's the communication domain. So clearly communication was the area of greatest concern. So conclusions, all the individuals who completed in-person phenotyping met criteria for a diagnosis of a motor speech disorder. Receptive and expressive language skills were at about the same level for most individuals. There was better nonverbal compared to verbal IQ, and significant global motor impairment was a common finding. I think this has clinical implications. So children who have a known pathogenic variant in MED13L early on, even before they're verbal, would benefit from early referrals to speech therapy. So we know that these kids are at risk for apraxia in particular, but also dysarthria. And so there should be a low threshold of suspicion for these types of disorders. So we should get kids into early intensive speech therapy, use evidence-based principles of motor learning before someone is certain that it's apraxia or not. I think we should make that assumption. It's the safest assumption. And then early on too, I think AAC strategies should absolutely be considered. So AAC refers to augmentative and alternative communication. A lot of kids can benefit from things like a speech generating device, and even kids with MED13L who are older and verbal. I've met many of you guys here. It can be really helpful just to improve functional communication when speech intelligibility impedes conversation. So I think this is a really important part of the therapeutic plan for kids who have MED13L. Okay, and that's everything, and take away the questions until later in the Q&A session, but I just wanted to say again, thank you so much to the families today. Thank you.